Converse.
Hello, hello.
How are you?
Well, okay for now.
Okay,
Yeah.
okay. I
Yeah.
got a question. Can I
Yes,
ask you
ma'am.
a question?
Yeah,
This is Cherie Harris. I need just a little more time on my lab, if that's possible.
that's fine. Just-
Okay.
yeah, just let me know.
Can I get it like a couple more days, like next Wednesday? Can I turn it in?
Sure,
I'm quite sure the class agrees with me if we
that's
could get a
fine.
little more time.
I'll bump it. That's fine. I got no problem with that.
Okay, all right.
Yeah,
Thank you.
I'll slide that over.
Okay, thank you.
Okay. Yeah. All right. Now,
Professor Convex,
yes, sir.
good afternoon. This is Joy. You see how the women, when they need something from you, they are so nice, right?
Yeah. All right. Okay, we're gonna mute now. All right. So, um, what we're dealing with today is the mode of action of these antimicrobial drugs. All right. Remember that the microbes consist of bacteria, fungi, protozoa, helminths, and viruses. Right. And one of the things that we understand about antimicrobial drugs is that ideally, for the drug to have the best possible outcome for the patient that has the infection, you want it to be exclusively toxic to your target, the pathogen, and to not have any adverse effects on the patient. And that gets harder and harder to do the more similar the drug, or the more similar the pathogen is to your own biology. All right. And so this is one of the reasons that it's relatively easy to come up with an antibiotic against bacteria, but it's tougher to get an antifungal. It's tougher, even more so, to get an antiprotozoal, and it's even tougher to get an antihelminthic. And antivirals are just really hard because they're using your own cells to reproduce and fight against you, right? And so it limits potential targets for drug action. Okay. The goal is to hit the bad guy and leave you relatively unscathed. Okay. A microbiocidal will kill microbes, but can also kill your normal flora. Remember that in the hotel body, you've got mostly good guests and a few bad actors. Those are the pathogens. And if you knock out the normal flora, you can give the pathogens a chance to get a foothold and overgrow, right? A microbiostatic reversibly inhibits growth. Okay, so that's different. It doesn't kill the microbe, right? An agent can be microbiocidal at a certain concentration and may only be microbiostatic at a lower level. One that is lethal to one type of organism may only inhibit another. Okay, so again, you got to pick your targets carefully. Antimicrobials use a lot of different mechanisms of action to either kill or inhibit microbial growth. The mechanisms are varied and can target highly specific structures or metabolic functions necessary for the survival of the microbe. They can be split into 5 categories: cell wall synthesis inhibition, protein synthesis inhibition, nucleic acid synthesis inhibition, cell membrane disruption, and metabolic antagonism. Now, bottom line here, okay, is that any pathogen that has a cell wall, that is an ideal target for a drug that would hurt them and not bother us at all. So here we're talking about bacteria and fungi, okay? If we can go after cell wall synthesis or cell wall integrity, then we have a really excellent chance to cripple the pathogen and not bother us at all, because we don't make cell walls, okay? Another important target is the ribosome, right? The bacterial ribosome is functionally identical but structurally different than our own. Okay, so if we go after that, it's more likely to hurt the bacteria than it is to hurt us. Now, let's be honest, at high enough dosages, we could have an impact on protein synthesis in mitochondria, since their ribosomes are similar to bacterial ribosomes. But that is probably an order of magnitude a higher dose than would be necessary to kill the pathogen. Okay. The weaknesses of the peptidoglycan layer, it's vulnerable to lysis through the action of penicillin and cephalosporins. Most bacterial cells have a cell wall composed of peptidoglycan, protecting it from rupturing in hypotonic condition. Young growing cells constantly manufacture the wall. Agents such as penicillin and cephalosporin react with the enzymes that do that and weaken the wall and make it more likely to lyse. Okay, so you can see there how it is a combination of carbohydrate and protein, but the production of these proteins does not require ribosomes. Okay, this is a unique way of manufacturing the cell wall. Okay, so if we go after what assembles this, then we have selective toxicity, and that is a beautiful thing. Okay, disruption of translation at ribosomes is another mechanism, right? Ribosomes of eukaryotic cells are not affected by agents that go after prokaryotic ribosomes, and as a result, there's a selective target for you, right? The specific chemical reactions that disrupt the translation of message may vary and include prevention of the initiation of synthesis, blocking the formation of peptide bonds between amino acids on transfer RNAs, and preventing the ribosome moving down the message. Whatever the specific point of attack, the result is the same: inhibition of the synthesis of the proteins. Antimicrobial agents that affect bacterial protein synthesis include tetracyclines, chloramphenicol, macrolides, and others. And so you can see here the prokaryotic ribosome, okay, 50S, 30S to make a 70S. We have all of these potential places where we can attack, right? We can attach to the ribosome itself and prevent it from moving forward. We can cause the ribosome to misread its message, okay? We can block ribosomal assembly on the message. There's all sorts of mechanisms to do that, okay? We can inhibit peptide bond formation, okay? We can inhibit transfer RNA binding, all right? There's all sorts of antibiotics that will do that. Disruption of nucleic acid synthesis is another potential target, okay? The complexity of metabolic processes leads to disruption at a lot of different points. Disruption of the synthesis of nucleotide components is one way to attack. Inhibition of DNA replication and interference with RNA transcription are other ways to attack. The end result is the same. An interesting type of mechanism that's employed by sulfonamides and trimethorphan involves mimicking the normal substrate of an enzyme using competitive inhibition. So what these guys do is they bind the active site, but then the enzyme can't do anything with it, okay? When these agents are supplied to the cell in high concentration, they substitute for their actual substrate for the enzyme, and so the enzyme can't do its job properly, okay? Disruption of the plasma membrane is, it's not as selectively toxic as these other targets, okay? Most membrane disrupting agents target different types of lipids in the cell membrane, which lends their specificity for a particular microbial group. Because all microbes share the same basic plasma membrane structure, these agents are useful against both bacteria and fungi. Unfortunately, they can be toxic to us, okay? So we can affect transport in and out of the cell. We can weaken the cell's integrity. We can affect prokaryotic and eukaryotic cells. So these are things that can act like detergents, or they can poke holes in the membrane. There's all sorts of methods to do that. We can also disrupt cellular metabolic processes by disrupting enzyme activity and production, disrupting essential metabolic compounds. These agents that break up these pathways are involved with the synthesis of proteins and nucleic acids and can be disastrous for the metabolism of the cell. So the idea here, right, is that you can interfere with the enzyme's ability to do its job by essentially not allowing it perhaps to bind its substrate, or causing the reaction to be inhibited, okay, by say modifying the active site. All right, that's another potential way to do it, or through this inhibition mechanism that we were just talking about. So those are all possibilities. Okay, those are all possibilities. When we look at the characteristics of antimicrobials, right, we've got all of these components to look at, right? How wide is the spectrum of activity? Does it hit everything, or does it just hit a few things? Is it selectively toxic? Is the target unique to the pathogen? Is it a microbiocidal? Does it kill the microbe, or does it just keep it from growing? Can it get to the site of infection in the effective dose? What is its solubility? Is it lipid soluble? Is it water soluble? How long does it remain active before the body breaks it down and clears it? Okay. What is the degree of resistance in the pathogenic population? Does it help our host defenses? Can it produce a hypersensitivity, an adverse event? Okay. How long does it last, and how affordable is it? Okay. A broad spectrum antibiotic is effective against a large variety of microbes, while a narrow spectrum is targeted against a much smaller population of microbes. Narrow spectrums are used once we know the susceptibility of the pathogen in order to keep the normal flora relatively healthy. All right, we don't want to hurt your good guys at the expense of going after the bad guys. Okay, the spectrum in the media, it can be effective against some gram-positive and gram-negative bacteria, but not all of them. Okay, so that's medium spectrum. So those are the ranges against which the drug is effective. Almost most antimicrobials don't inhibit the growth of all pathogens. Drugs have a spectrum of activity, which is the range of the pathogen types that it's effective against. Advantage of using a broad spec is the high possibility that's going to be targeting the pathogen. The disadvantage is you wipe out your normal flora, and then the pathogen could grow back afterwards, right? This leads to a phenomenon called superinfection. An example, okay? Let's say that you're taking a broad spec antibiotic for an infection, and you knock it down, right? But in the course of doing that, you wipe out the good bacteria in your colon, okay? And then when you're off the broad spec, there's a few cells of C. diff in there, and bang, they take off, okay? And now instead of having your normal flora, you have C. diff infection, okay? And that's one of those things that happens sometimes. In selective toxicity, we just go after a target that's unique to the pathogen, and we don't harm ourselves much, okay? The toxic dosage and the therapeutic dosage are way spread apart here, okay? So you don't have to worry with the therapeutic dosage having an adverse effect on the patient, okay? So the idea here, right, is don't hurt the patient, hurt the pathogen. Microbiocidals.
So, Professor,
Yes, sir.
Professor Canvas, so
Yes.
when do you know which of the doses, I mean, the toxic doses compared to, I mean, how will you figure out
Okay,
the
so that's
toxic
a good
doses?
question, all right? And we're actually going to see a type of test called Kirby-Bauer, where you can determine what we call the minimum inhibitory concentration against a particular pathogen before you use it. So generally, the way it works is you go in with an infection, and the doctor will, just to deal with it right away, he'll start with a broad spec, but then they'll do testing on culture from you to find out what drug is best against the pathogen. And the way that works is through a test called Kirby-Bauer, which I'll show you in a bit. And then you switch the prescription from the broad spec to this narrow spec. And then you're able to wipe out the target, right? So you start with an assumption, which is we want to get the infection down, okay? But then we switch off to something that's going to be narrow spec so that you don't build up resistance to, or the bacteria don't build up resistance to the broad spec, the bacteria that are growing in your body, the potential pathogens. So it's a sort of a two-step thing. Because of the potential negative effects of drugs on the host, the use of a killing agent instead of an inhibitory one is not always a simple decision. Microbicidals can also kill the natural flora in areas such as healthy intestine and create the potential for a GI problem because you've wiped out the inhibitory effects of the normal flora, right? Can it get to the site where it needs to be active? Yeah, question.
Yeah, Doc, because I mean, before, unless it's prophylaxis treatment, I believe the lab's result will indicate which antibiotic each virus or bacteria is susceptible to, right? But my
Right.
question of what I'm a little confused about is the toxicity level. Voice, that's the part I was.
Okay, so when we're all right, so that's data that's already known. Okay, so when you have I don't know, let's just make up a drug X, right? When you have a drug, right, you're going to have what we would call the therapeutic dose and the toxic dose. Okay, this is the range for what's called LD50. Okay, and this is ED50, effective dose 50%, right? And this is published data. Okay. Lethal dose 50 is the dose, so this would be the concentration. Okay. And this would be, no, I'm sorry, this would be the concentration down here. Okay. And this would be the number of patients affected. Okay. So you see that the LD50, actually, so first. There's a separation between these two ranges. Okay. Such that the maximum dose for the ED50, okay, is not going to overlap with the lowest dose of the LD50. Okay. If you were to have a drug that had a different profile, right, where the LD50 and the ED50 overlapped, concentration, okay, number affected, okay, then the therapeutic index here is much tinier, right? It's almost non-existent, right? No, let's say low therapeutic index, okay? So here you have a higher therapeutic index, right? So if we can do it, if we can use it, we would always pick a drug like this versus a drug like that, okay? And we would have information regarding, once we know what the organism is, right? Then it gets even clearer for our choice, right? So you might start out with a broad spec that has a high therapeutic index, okay? And then you would go to a narrow spectrum drug. Broad spec. Then to narrow spec. But in each case, you want the highest therapeutic index. Okay, remember the therapeutic index is just the ratio of the LD50 to the ED50, right? You want that to be as big a number as possibly can be, okay? Because otherwise you're going to have toxic effects for the patient. Now you have to consider the patient too, right? What's going on with the patient is going to determine your choice of drug as well. For instance, if they have compromised liver or kidney function, then the choice gets even tougher, right? And you're limited in what you can use. And sometimes you just have to take a risk, okay? But you have to tell the patient why you're doing what you're doing, okay? In order to be effective, the drug has to be easily and effectively delivered to the site of infection. As a result, the important criteria is the efficacy of the drug and its ability to cross biological barriers and get to the target. Barriers also may be introduced by inflammation and disease, abscesses, exudates, necrotic material. All are barriers, okay? Impeding the ability to get the drug to the pathogen and do its thing, okay? If the antimicrobial drug isn't sufficiently soluble or becomes too dilute in solution, the effectiveness is greatly diminished. Also, if the delivery or transport of the agent doesn't efficiently and rapidly deliver the antibiotic to the site of infection, the drug won't have the opportunity to fight the pathogen, even if it's in solution in the correct concentration. Okay, so all of this stuff we have to consider. Right? The time of activity, right? The duration of action of time is a major consideration when evaluating the effect of a drug in an infection. If the destruction of a population of pathogens requires sustained concentrations of an agent over a specified period of time, any shortening of that time significantly decreases the effectiveness of the treatment. By subjecting the organisms to the agent for a time that proves to be non-lethal to many of them, the risk of producing a resistant population is increased, right? Because what you're doing there basically is selection. Right? You're doing selection. Okay, you're selecting for those in the population that have the greatest potential for resistance to your drug. Okay. Ideally, you don't want any antimicrobial resistance, but that's difficult to obtain, right? Genetic mutations produce resistant strains all the time, okay? Acquisition of resistance by the different methods of transfer that we talked about, right? And we should include in here transposition. In addition to transduction, conjugation, and transformation, right? 4 ways that they can pick up resistance gene, right? It's the nature of the agent and its mechanism such that the population of pathogens is less likely to develop resistance to the agent. Its effectiveness and scope of use would be dramatically increased, right? So if something can mutate out resistance to a pathogen at a high rate in a particular organism, it's not worth using, right? Whereas if it's difficult for those mutants to pop up, then that's a better choice. Okay, but remember that drug resistance among bacteria spreads rapidly. Okay. All right. It has to help our own defenses. When layers of defense are compromised, antimicrobials need to be administered. Foreign substance introduction results in risk of upsetting the balance of defenses, inhibiting the body's natural response. Normally, natural defenses in our body, including barriers and portals of entry, and protective cells, and environment within the host, and an acquired immune system, both active and passive, do an excellent job of fighting off infections in the healthy host, right? We talked about the fact, well, we've spoken about the fact that you've got really 4 levels of protection, right? First level are really, your good microbes. Okay. Then your skin and mucous membranes. And their secretions. Then the action of your granulocytes. Okay. And inflammation. And finally, the lymphocytes and macrophages. Okay. This is what's called our adaptive immunity. Adaptive. Okay. And then this stuff really, these two, you think of them as innate immunity. And they all work together and use different weapons to keep you healthy, right? What we don't want is for the administration of some kind of drug to compromise any of these levels of protection. Right? As an example, you take a broad spec antibiotic for an extended period of time, you could lose your good microbes, right? Bang. So now we've lost one of the things that keeps us normally healthy. Okay. So you pick carefully, right? Certain antimicrobials produce hypersensitivities. Okay, these are called drug allergies. Okay. Patients with drug allergies, such as allergies to penicillin, have to be careful to tell the medical personnel about their allergies, or they could end up in shock that can result from receiving penicillin that could be lethal. Okay, so this is why you have to take a patient history. Right? Otherwise, you make a goof that you didn't anticipate, right? If the patient says they don't know whether they're sensitive or not, then you play it safe. And you pick a drug class that has less reported incidence of allergic response. Okay. But any drug, it doesn't matter what it's for. It doesn't matter if it's an antimicrobial, doesn't matter if it's to control blood pressure. There's going to be in the population individuals that are allergic to it. All right. That's just because we have biodiversity. Okay. And if we have biodiversity, that means in every human population, there's going to be some individuals where you give them that compound and they don't feel well. Okay. It even happens with vaccines, right? You give a vaccine and certain people will have what's called an adverse event. Okay. Well, you know, did the vaccine contain, a pathogen? And the answer is no. They had an allergic response to what was in the vaccine, a hypersensitivity. And so their immune system produced the symptoms that you associate with that, inflammation, fever, aches, and so on. Okay. Everybody has their kryptonite somewhere, right? Because our adaptive immunity is functionally identical in everyone, but it's structurally different. Since it's made of different parts, there's going to be something out there somewhere that's going to cause you to have a hypersensitivity, most likely. Okay. It's just that people that have allergy problems all the time, they run into it all the time, right? Whereas a lot of people, they just don't. Okay. Shelf life and stability, right? You have to be able to store the thing and it has to be able to keep. Okay. If we can't do that, we got trouble, right? The longer the shelf life, the more easily the agent can be stored and made available. And it has to be safe, right? In addition to logistical and economic advantages of a long shelf life, there might also be a safety consideration, right? Chemical breakdown results in antibiotics with either low potency or chemical products that degrade and become toxic, right? So this is one of the unfortunate things that's starting to happen around the black market, right? Y'all know you can get drugs in ways other than through your doctor, okay? You can go on the internet, you can get these drugs from other countries, and they come in and you have no idea, all right, whether it's the drug that you wanted, whether it's an old batch of the drug, whether the drug has broken down over time, you just don't know, okay? A lot of those drugs that are distributed by these other places are actually excess drugs that were sent to these distribution warehouses from patients that either wanted to sell them, right, or no longer needed them, which means that in many cases they're too old, okay? And that's going to cause trouble, all right? Bottom line, right? You should do what it says on the label when you're prescribed a drug, right? Like, for instance, with an antibiotic, you should take the entire course and then throw the bottle away, all right? If you keep it, you're not really helping yourself. You're not really saving money, because what's happening is that the drug is becoming less potent and may, in some cases, become toxic, right? So you just don't want to do that. You just don't want to do that, okay? It has to be affordable and available, right? It's obvious that even the most effective agents won't be useful if the product isn't widely available to patients in need. And once available, it's also important that the product be affordable, okay? Now, how do we determine if this is going to do what we want it to do, right? And you asked me this question earlier, right? How do we know what drug to pick, okay? So there's a variety of methods, right? There's the Kirby-Bauer or disc diffusion method, there's the MIC method, and there's the serum killing method, all right? So with the Kirby-Bauer method, right, what you do is you take your organism of interest, you streak it out on nutrient agar and grow it up in a lawn, all right? And then you put discs impregnated with the particular drug on the lawn at the same time, and the drug in the disc diffuses into the nutrient media and kills bacteria within a certain radius, okay? And you evaluate whether it's effective based on the radius of killing, all right? So let's take a look at this, all right? I've got this down here. I should have had it down here. Did I not store it? Well, I'll pull it up. Let's discard this. Take a look how this works. It's not as simple as you just look at the zone of clearing and say, well, the biggest one is going to be the most effective drug. Okay, it's a little bit more subtle than that, okay? Because drugs have different sizes and different diffusion rates.
discs are marked. In this case, you might read it. This one says BA30, which stands for 30 micrograms of glass. We're going to put these on the plates. And 2 normally shows up in the dispenser. Right? Dispenser holds 6, 3 on this side, 3 on this side of the dispenser. When pushed, this will dispense a set of filter paper discs on the plate. Take a plate, take an organism, flame your loop, cool your loop, and spread the organism over the entire surface of the plate. Going until you cover the plate. You'll notice that I've turned this multiple times, going back and forth. Go ahead, don't talk on the plate while you're doing it. Then flame your loop, cool your loop. That part's done. Right? Once you're done with that, put the lid down and write on the plate what the organism is and your initials. Do it now because once you get the filter paper discs on, you gotta leave it right side up. Don't leave it right side up, discs fall off. If discs fall off, it doesn't work so well. Once you're done with that, discs. Right side up. Lid off. Set our dispenser right over the top. I took the lid off. Otherwise, the disc just wind up on the lid and does no good at all. And I just simply push down. Release. That's it. Put back on. See our discs sit on. Side up. This one here. Notice that we don't have anything growing except for one big fuzzy, one big mole growing on. Well, one big mole growing on this helpless function. It's like it's not inoculated at all. It's a demo like the other one. The thing is, all of these are antibacterial antibiotics, and obviously, considering the fuzzy grown right on top of the disc, they don't work so hot on fungus. So, in order to do something against fungi, we would have to do this with a bunch of antifungal discs instead of antibacterial discs. One winds up looking like this. What you'll notice is, in this case, this one that says AM10 or ampicillin 10 micrograms, it doesn't affect it at all, does it? It's resistant. And if you go around, you can see slightly bigger areas clear on the outside. Clear areas of zones of inhibition, as they call them. And one would think that the bigger it is, the better it is. Not necessarily. Some of the antibiotics are smaller in size than others. The smaller they are, the better they come out from the disc out through the So sometimes the bigger zone doesn't mean smaller. Maybe this one happens to be a very big antibiotic and it actually works better than this one here, which might be a very small antibiotic. But in order to determine whether the organism is resistant or sensitive, simply measure the distance, disc across, and then compare it with a book what the standard is supposed to be. Antibiotic sensitive or resistance. Same way. So you may not be doing this in a lab, but put one sheet out on this. And like the rest of what's going on.
So that's literally how you do it. Okay. The information that you gather from the plate is compared to a chart, right? And that chart tells you whether the drug is effective, is ineffective, or is intermediate. All right. So you literally measure the zone of clearing, and then you go to a chart that will give you a cross reference for the drug, and in many cases the organism, right? And then you compare the diameter to what you pull off the chart, and then that determines your choice of drug. Okay. So literally how they do it. Okay. So Kirby Ballard, right? Let's go. So that's how you determine. All right. Another way of doing it is a little bit more sophisticated. All right. It is called the minimum inhibitory concentration method. All right. And so the idea is to find the lowest effective concentration of an antibiotic by looking at how turbid, how cloudy the media is, right? Lack of turbidity signifies inhibition of bacterial growth or killed microbes. The minimal bactericidal concentration is the lowest concentration containing no organisms, right? So that's how you do the mix. Now, you can do it with turbidimetry. Sometimes you can use a colorimetric reaction in order to do this. And there's like a plate reader. You can do it in a 96 well plate, and the plate reader will pick up the change in color, indicating growth or no growth. Okay. Serum killing power uses the patient's blood and extraction of the serum from that, the bacteria added to the serum, and we see what happens, right? A bacterial suspension added to serial dilutions of patient serum to determine if the bacteria grow in the tube of serum indicates that the antibiotic is effective, whereas no growth indicates that it's not effective, right? So you vary the concentration, you keep the amount of serum constant, right? And then you see where that number is, all right? Now, we talked earlier about, the potential for what we call adverse effects, right? I mean, that's a thing, okay? Potential organ damage, we can see hepatotoxicity, we can see problems in the GI, we can see nephrotoxicity in the kidneys. It can affect the cardiovascular system and red marrow. It can affect the nervous system, so neurotoxic, respiratory system, the skin, okay, and it can affect the bones and teeth. All drugs can be potentially poisonous, and the administration of a therapeutic drug can possibly lead to side effects that can be serious. The administration of drugs involves previous assessment of risks and benefits. This type of assessment is called the therapeutic index, right? So every drug, we were talking earlier about this, right? Every drug, period, anything that you ingest, right, has an LD50, okay? Even water, okay? LD50 means lethal dose 50%. Okay? Every substance on the planet has something. It has this number, okay? And what you want to do always is pick something that has an LD50 that's very, very high, okay? So that you don't have the remotest chance of killing your patient, okay? The LD50 for water is very, very high. The LD50 for carbon monoxide is much lower, okay? Just to give you a relative idea of how these numbers are assigned, okay? Now, resistance to antimicrobials happens as a result of spontaneous mutation, but also gene transfer, right? And let's again put in transposition here too. Transposition. Stop that. Silly button. Transposition. Okay. The major challenge today is the problem caused by bacteria that are already resistant to antimicrobials, as well as those that acquire resistance because of all of these different mechanisms, right? The evolution of bacteria towards antibiotic resistance, including multi-drug resistance, represents an aspect of the general evolution of bacteria and most likely can't be stopped, right? So as illustrated here, right, in the figure below, as drug exposure increases, non-resistant cells are eliminated. Eventually, we only have resistant cells, right? That's just, again, all you're looking at here is selection. Okay. Natural selection. All right. Now, so you might ask, oh my God, what are we going to do? Stop using antimicrobials? And the answer is no. You switch what you use. Okay. You might be using second or third generation antibiotics, and maybe you switch back to first generation. Okay. Until those become less effective. And then you switch back to second generation, and you keep moving around and you keep looking at the statistics in order to inform you, what particular organism out there is susceptible to, right? If you take selection off, all right, let's take this experiment here one step further, right? If you remove the selection, remove the drug from the media. Right? What will happen after several passages is that we'll actually go back to something that's more like this, okay? Where you've got resistant and non-resistant organisms in the population, okay? Because you've pulled the selection off. And you can do the same thing out there in the ecosystem, right? Remove the selection, the composition of the population will change. Okay? What are some of the mechanisms of resistance, right? What do these genes do? Well, change membrane permeability to the drug, increase the elimination of the drug, and literally pump the drug out of the bacterial cell. Change in the target, right? Change in a metabolic pathway, change in a previously inhibited enzyme, development of defensive enzymes, right? All of this, okay, is genetic. All of this is genetic, all right? Multiple resistance can happen, right? You can get microbes that are resistant to more than one drug at the same time. Multi-resistant strains occur in hospitals, nursing homes, and other clinical settings. In these environments, the constant use of different types of antimicrobials eliminates sensitive cells while encouraging the development of resistant ones. Superbugs, these multi-drug resistant organisms, are an example of this, right? At present, multi-resistant organisms include strains of staph strep, enterococcus, pseudomonas, mycobacterium, and even the malaria-causing protozoa plasmodium. In addition, resistance to only one type of drug, unfortunately, also may allow for resistance to a similar drug, and that's called cross-resistance, right? So you can imagine, let's just take an example, right? Let's say that you've got bacterial cell A, and he is TET resistant, tetracycline resistant, okay? Because he has a little TET-R plasmid in there, okay? A piece of DNA with TET resistance on it, in addition to his chromosome, okay? And then you have another cell, right? And let's say it's ampicillin resistant, okay? Ampicillin resistant. Okay. And so he has an AMPAR plasmid on it. Okay, in addition to his genetic material. Well, what they can do, right, is of course they can grow a pilus between them. We talked about pili, right? These are hollow tubes that allow us to transfer genetic material, right? We grow this pilus between these two cells. And we could transfer a copy of the genetic material in either direction, right? In either direction. So we could send the AMP plasmid that away, or we could send the TET plasmid this away. And then what happens when we're done? We have a bacterial cell that's resistant to both drugs, okay? Even though the AMPR has never been exposed to tetracycline and vice versa, all right? And that's why it spreads so fast. We have forms of venereal disease now that are resistant to up to 50 different drugs at the same time, okay, because of this phenomenon. So how do we prevent, right? Well, there's hand washing, eliminating unwarranted use of antibiotics, right? Targeting narrow range of microbes, using drug combinations for synergistic effects, okay? Isolating facilities with ongoing infections, looking at current data. Antibiotics should be taken as prescribed, and that is called compliance. Okay. The patient has to do what the doctor says. Okay. All right. A patient needs to be compliant and honest. Okay. And sometimes that doesn't happen. Leftover meds should be discarded. Don't take anyone else's scripts. Use antimicrobial soaps and lotions. Prevent disruption of normal flora. Avoid selecting for resistance. Since antibiotics were discovered more than 50 years ago, the microbes have developed resistance to many of the agents that are on the market. It's become a worldwide public health problem. In '96, the National Antimicrobial Resistance Monitoring System was implemented to track the emergence of antimicrobial drug-resistant organisms and strains. The WHO has provided a framework of action for effective containment at various levels. Right? Antibacterial agents, right? These are natural, they can be natural antibiotics. For instance, penicillin is a natural product of fungi, right? Penicillium mold, right? Semi-synthetic antibiotics, which are chemically modified natural antibiotics. These are often called derivatives. Derivatives. Okay. And then synthetics, which are totally made in the lab, all right? Natural antibiotics have the capacity to either destroy bacteria or inhibit growth. Semi-synthetics are created for efficacy. Synthetics can act identically to antibiotics, okay? So here's a little bit about how these drugs work.
My name is Suringan, but I'm making some videos. Please make sure you subscribe, join the forum group for the latest videos, places, and Facebook. My name is Suringan. In this video, we're going to talk about the mode of action of antibiotics. So we're not going to look into extreme detail, but we'll just look at the overview. So mode of action of antibiotics. Now we have to know that antibiotics either destroy or slow down the growth of bacteria. So therefore, if it destroys a bacteria, it is bactericidal. If it slows down the growth of bacteria, it's bacteriostatic. Here I'm drawing a typical bacteria with cell membrane and cell wall. The cell wall and cell membrane structure I'm drawing is characteristic of a gram-positive bacteria, but we will just assume it is any type of bacteria. Within a cytoplasm, within the bacteria, we have a circular DNA. We can have RNA being synthesized and ribosomes here. Ribosomes will synthesize proteins. The first type of antibiotic are the ones that I want to talk about are the ones that target the cell membrane. These antibiotics will disrupt the cell membrane function, which means that they disrupt the phospholipid bilayer. So here is the cell membrane, and here is the cell wall, known as enteroglycan. they are different. The antibiotic binds here to the cell membrane. It will alter the cell membrane structure and will make it more permeable. This will disrupt the osmotic balance, causing leakage of cellular molecules, and essentially it will increase the water uptake, leading to cell death. Examples of these drugs are polymyxins, which I spelled wrong, and polyenes. Now, polyenes are actually specifically used against fungal pathogens. But that's just a note to know that you can use polyenes that target some membranes in fungus. So polymyxins and polyenes target cell membranes. Other antibiotic classes target the cell wall. These antibiotics will inhibit cell wall synthesis, so these drugs are therefore bactericidal. They will cause bacteria to die. Example of these drugs are penicillin, cephalosporin, and bacitracin. All these drugs have different mechanisms of action when it comes to inhibiting the cell wall synthesis. A good thing about these drugs is that it won't affect human cells because human cells do not have peptidoglycans. We do not have cell walls. Penicillin and cephalosporin are beta-lactams. And then you have the bacitracin and other glycopeptides that prevent the synthesis of cell walls as well. Antibiotics that disrupt the cell wall is pretty extensive, and so hopefully I will make a video on that. Cell walls are important to bacteria. Disrupting the cell wall or preventing cell wall synthesis will result in cell death. The next type of antibiotic class is the ones that can inhibit here DNA synthesis, or what's occurring here, RNA synthesis. So these antibiotics will inhibit RNA and DNA synthesis. So here the bacteria are typically DNA replicated, of course, like so. So we have antibiotics that prevent DNA from being synthesized. These are quinolines and also a class known as nalidixic acids. And then, of course, RNA can be made from the DNA. And there are drugs such as rifamycin, which prevents RNA synthesis and therefore protein synthesis. Other classes of antibiotics also inhibit protein synthesis itself. Protein synthesis is carried out typically by ribosomes, which translate mRNA into proteins. Bacteria need to make proteins in order to survive. However, there are antibiotics that target either the 30S subunit or the 50S subunit of the ribosomes. Erythromycin and chloramphenicol target the 50S subunit of ribosomes, whereas there are tetracycline, streptomycin, and gentamicin that target the 30S subunit. Either way, this will disrupt the ribosomes from making proteins, which will result in the bacteria obviously unable to make proteins. So the bacteria is pretty much useless. Thus, the bacteria can still be alive, but it just is unable to make anything. And so these antibiotics that target protein synthesis are bacteriostatic. They prevent bacterial growth. Last type of antibiotic are the ones that inhibit folic acid metabolism. So normally, this green thing here called PABA, which stands for para-aminobenzoic acid, it's a precursor to folate. So the bacteria can metabolize PABA into folate. There are antibiotics out there that inhibit folate or folic acid metabolism. See, PABA normally can be metabolized to folic acid within the bacteria. And folic acid or folate is essential for the synthesis of adenine and thymine. two of the four nucleic acids that make up DNA. These antibiotics, such as sulfonamides and trimethoprim, prevent conversion of GABA to folate and therefore prevent proper DNA synthesis inside. Humans do not synthesize folic acid, and so these antibiotics are okay to take for humans and are selective towards bacteria. I hope you enjoyed this short video on antibiotics. Thank you for watching.
Sulfonamides, first antimicrobials developed that result in metabolic disruption of bacteria. Trimethorphan, used in combination with sulfonamides, quinolones, broad spec, treatment of a wide variety of infections. They're human-made drugs and therefore not considered to be antibiotics, but synthetic antibacterial agents. Okay, let's take a look at penicillins.
Preventing peptidoglycan synthesis is detrimental to bacteria, but has no effect on host cell processes. Therefore, many antibiotics interfere with peptidoglycan biosynthesis. Cycloserine inhibits the conversion of L-alanine to D-alanine. It's the formation of the D-alanyl D-alanine required for peptide side chain formation. Bacitracin inhibits the pyrophosphatase that removes the terminal phosphate from bacterprenyl pyrophosphate. Without removal of the terminal phosphate, bacterprenyl cannot be used again to transport disaccharide pentapeptide precursors through the cell membrane. Vancomycin binds to the D-alanyl D-alanine portion of the pentapeptide side chain. This prevents formation of the covalent cross-links between adjacent glycan chains. The cell wall continues to grow, becomes weaker and weaker until the cell lyses. An enzyme called the transpeptidase forms the covalent bond between peptide side chains, which leads to the terminal D-alanine. Penicillin inhibits this transpeptidase. As the cell wall continues to be formed, the lack of covalent bonds between the peptide side chains eventually results in cell lysis.
So that's how the penicillins work, okay? The cephalosporins are very, very similar, right? Antibiotics are chemotherapeutics naturally produced by some microbes, such as molds, and even some bacteria. Semisynthetics, on the other hand, are natural products that have been chemically modified in the lab to improve the efficacy of the antibiotic and expand the spectrum of activity. Penicillins have proven to be relatively safe, although a small percentage of the human population have allergies. Each new generation of cephalosporins has improved dosing schedules and has fewer side effects. Now, an important point with all this, of course, is that you can develop resistance to anything, and you're probably familiar, let me should put this back one up here. You're probably familiar with a condition called MRSA. Okay. So MRSA stands for methicillin resistant staph aureus. Okay. What is methicillin? Methicillin is a derivative of penicillin. And it was developed because bacteria were figuring out how to destroy penicillins relatively easy. Well, not figuring out, they were being selected for penicillin resistant antibiotics. And so methicillin is a modification of penicillin meant to overcome that. And of course, what happened when we started using methicillin is that we selected for organisms that were methicillin resistant. Okay, so it just goes round and round and round. You have a weapon and then they develop a counter weapon. Okay. Tetracyclines are broad specs. They're both natural and semi-synthetic. and they inhibit protein synthesis, so they attack prokaryotic ribosome. Okay. Possible effects, side effects of TET include nausea, diarrhea, teeth discoloration, damage to liver and kidneys. Okay. We have to point out that we sort of talked about this before. All drugs are going to pass through the liver and the kidneys to be eliminated, right? And if you tax the liver too much, you can damage the liver. That's called hepatotoxicity, right? Aminoglycosides are naturally produced in bactericidal, and they can be toxic with many side effects. Resistance by some bacteria to streptomycin has already been noted, right? Side effects include deafness, kidney damage, loss of balance, nausea, and allergic responses. Macrolides are effective against gram-negative bacteria, mycoplasmas, and a few select gram-negative bacteria, and are useful when fighting infections in patients who are allergic to penicillin. They're also effective in the treatment of whooping cough, diphtheria, gastroenteritis caused by Campylobacter, and pneumonia from Legionella. Side effects of chloramphenicol can be severe and include allergic and neurotoxic reactions and irreversible bone marrow damage leading to fatal forms of aplastic anemia. Okay. So again, you know, you're not without damage to the host here, right? Bacillus antibiotics are narrow spec, produced by bacillus species. They're often topically applied, if they're bacitracin or polymyxins, for surface infections. They can be administered internally when the patient can be easily monitored because of the potential for kidney damage and respiratory arrest. So here, again, you're talking about something that if you take it enterally, right, you have to take higher doses, right? So here we can see the spectrum of activity, okay? All right, now antivirals, it's a whole different story, all right? They're limited in their effectiveness. They can damage the host. They target points in the viral life cycle that are unique to the viral replication mechanism, okay? They can prevent penetration, block transcription or translation. They can prevent the maturation of the virus, okay? The thing about this, though, right, is remember that the virus uses your own cell as a virus factory. And so if we attack viral replication, we kind of attack you, okay? And that makes it hard to come up with a lot of good antivirals, all right? DNA viruses, we don't have a cure for. We don't have an antiviral for, really, okay? Those would be, for instance, the herpes class, right? If you get them, you got them. They're in you for life, and there's not a lot you can do. So what do we look for? All right. We look for something in the viral life cycle that is unique to the viral replication cycle that we don't make, okay? And I'd say, well, like what? Okay, well, we can start with this one, right? Acyclovir is a guanine analog, okay? The polymerases that are used by retroviruses which, you know, they take RNA, they could create a DNA molecule from that information. That's called reverse transcriptase. Acyclovir can target that because the reverse transcriptase isn't discriminating enough to determine whether it's the drug or whether it's an actual building block. And when you incorporate the acyclovir into the growing DNA molecule, it stops, okay? So that is a unique target, all right? That is a unique target. Reverse transcriptase is not something we make, all right? So we can go after that. Amantadine is used to prevent influenza A, okay? It can be up to 80% effective, right? And again, if you're targeting here with most of these drugs, all right, you're targeting either double-stranded RNA, all right, in the cytoplasm, okay, or in the case of the reverse transcriptase, DNA from RNA, right? And so viruses that are RNA viruses, we have some tools for, okay, because RNA viruses do all their work except retroviruses. Okay, do all their work at the cytoplasm. All right. So we have a way to get at that. If it go into the nucleus, we kind of can't get to you. Okay, that's a problem. Okay. Azithromycin, again, for AIDS. I mean, it's the same idea that we talked about with acyclovir. Okay, it gets incorporated into the growing DNA made by reverse transcriptase. It stops the production of the DNA molecule, and then you're done. Okay. Okay, interferons. Now, this is an interesting story. All right, interferons they're natural products. Okay, they're natural products. What happens when a cell gets, so here's a cell, right? And then a virus gets in. Here comes the virus. The virus gets in. And it starts to take over, all right? And what the cell will do is it'll spray out interferon to the neighboring cells. Okay. The neighboring cells will be warned and they'll start whipping out a protein called dicer, among others. Okay, but dicer is one of the big ones. Okay, and what dicer does is it chops up double-stranded RNA so that that virus, if it lands in there and tries to do its thing, it can't do much. All right. So we thought, wow, this interferon, this is great stuff. So we started marketing interferon as an oral for treatment of cancer and treatment of viral infections. And what we found out was that at high doses, it's hepatotoxic. It's bad for your liver. So the cure is worse than the disease. So the lesson there is that the body makes small amounts of interferon for a reason, okay? And that reason is the body can't handle high amounts, okay? So we still give it in low doses, all right? But this is an antiviral. This is something that you give after you've got virus, all right? There just aren't that many of these things, okay? All right, now the next one is antifungals, all right? So let's look at some antifungals.
Professor, I got a question.
Before we get today's lecture started, please remember, click here to check out our brand new app-based NCLEX product, loaded with the highest quality NCLEX style practice questions, and complete with detailed video rationales that break down the questions for you. So finally master all the hard select all the right questions.
Plus, all our NCLEX memberships come included with our entire library of over 1,000 videos and study guides and teaching. Come see why over 100,000 students have invested in future.com. Click here to get started for free. Now for antifungals, think anti-fungus, mainly used to treat candida fungal infections. So first up, N-azole, like fluconazole or ketoconazole, given to treat male and skin candida fungal infections. So guys, we think nasal, think N or male fungus. Now it's taken for 2 to 6 weeks, and it's very liver toxic, like most antifungals. So the key point here is that it does not treat C. diff infections. Remember, nasal treats male fungus, and dazzle treats the gut or C. diff bacteria. Now, guys, don't get tricked with these sound-alike names. Nasal is not a dazzle. So metronidazole, like flagyl, treats C. diff. Remember, there's nothing dazzling about C. diff. Now, amphotericin B is for serious fungus and causes serious terror on the body with lots of side effects. So both HESI and ATI had questions concerning the adverse effects. And guys, renal injury is the biggest one. So write down these key points. A creatinine over 1.3 means bad kidney. 3 mLs per hour or less means the kidneys are in distress. And oliguria, low urine, just another name for it. Thanks for watching. For our full video and new quiz bank, click right up here to access your free trial. And please consider subscribing to our YouTube channel. Last but not least,
Professor, I can't hear it.
Oh, well, hold on.
All right, guys. See you next time.
I'll crank it up. Hold on. Let's get the other speaker up.